Question number 161. Linking of a foreign DNA with a vector became possible with discovery or identification of. So we need to find out which option is responsible for linking the foreign DNA with a vector. And the very first option given is DNA ligase. And this is the correct answer to our question. Because ligase, which is also known as molecular glue, is used to link different DNA fragments. The next option given is restriction endonuclease, and this is an incorrect option because restriction endonuclease, which are also known as molecular scissors, they are used to cut the DNA fragments and not to ligate the DNA fragments. So this is an incorrect option. The third option is DNA polymerase, and this is also an incorrect option because DNA polymerase is an enzyme which is responsible for the synthesis of DNA. The last option is exonuclease, and this is also an incorrect option because exonucleases are the enzymes which will cleave the DNA sequence at its terminal ends. So the correct answer for this question is option number 1. Let's proceed to the next question. Question number 162. First clinical gene therapy was given to treat ADA deficiency. Which of the following vectors was used to transfer ADA gene into lymphocytes? This is a direct question, and the correct answer to this question is retrovirus. This retrovirus is a disarmed vector which is used to transfer this ADA gene into lymphocytes. On the other hand, the other three options like bacteriophage, plasmid and cosmids are incorrect options. Bacteriophage is a virus that infects bacteria. Plasmid and cosmids are extrachromosomal, self-replicating closed circular DNAs, and they are not involved in gene therapy. And they are not the first vectors which are used for gene therapy to treat ADA deficiency. So the correct answer for this question is option number 1. Let's proceed to the next question. Question number 163. The figure below is the diagrammatic representation of the E. coli vector PBR322. Which one of the given options correctly identifies its certain components? The very first option given is A, codes for the antibiotic ampicillin, and in this given diagram, A represents what? A represents antibiotic resistance gene for against ampicillin. So it's an ampicillin resistant gene. So option 1 is an incorrect option because A doesn't code for ampicillin but for ampicillin resistance gene. Option number 2 is D determines the copy number of plasmid, and D, which is this, it codes for rope. And this is an incorrect option because it is this E which codes for ORI, which determines the copy number of plasmid. So this is an incorrect option. The third option is digesting the plasmid with the restriction enzyme having C as the recognition site will generate sticky ends, and C is here, which stands for ECO R1 restriction endonuclease site. And if we'll digest the plasmid with the restriction enzyme ECO R1 at this site, it will generate sticky ends. And this is a correct option and is a correct answer to our question. Our last option is introducing foreign DNA in B will make the recombinants resistant to tetracycline. And this B, which is tetracycline resistance gene, if we insert the alien DNA over here, this will lead to insertional inactivation of this tetracycline resistance gene and will make the host cell susceptible for tetracycline and not resistant to tetracycline. So this is an incorrect option. So the correct answer for this question is option number 3. Let's proceed to the next question. Question number 164. Select the vectors which have the ability to incorporate a foreign DNA in chromosomal DNA of plant cell. So you need to find out the vector with respect to plant cell. And the very first option given is disarmed tie plasmid. And this is a correct answer to our question because it is disarmed tie plasmid which is used to incorporate foreign DNA in chromosomal DNA of plant cell. The type plasmid from agrobacterium tumefaciens it is a bacterium which generally attacks dicot plant. And this natural infection of this agrobacterium tumefaciens, which will lead to the incorporation of foreign DNA in chromosomal DNA of these plants. So it is this disarmed type plasmid which is used to incorporate foreign DNA into plant cells. Let's discuss other options also. Second option is PBR322. It's an extra chromosomal autonomously replicating DNA which generally doesn't incorporate into the chromosomal DNA of host cell. So this is an incorrect option. The third option is bacteriophage. Bacteriophage is a virus that infects bacteria and not plant cells. So this is an incorrect option. And option number 4 is also an incorrect option. So the correct answer for this question is option number 1. Let's proceed to the next question. Question number 165. All of the following drugs are used to treat insomnia except. So you need to find out which drug is not used to treat insomnia. And what do you mean by insomnia? A person suffering from insomnia faces difficulty in falling asleep at night. Now drugs that suppresses the activity of CNS or brain are used to treat this condition called insomnia. And among the given options, barbiturates, benzodiazepines and Valium, these are the drugs which are used as sedatives or depressants, which will depress the brain activity and produces the feeling of calmness, relaxation, drowsiness and deep sleep. So they are used to treat this condition called insomnia. So these cannot be the correct answers because they are used to treat insomnia. On the other hand, the last option, cocaine, is the correct answer to our question because cocaine is a stimulant which makes a person more wakeful and alert. So cocaine can be used to treat this condition called insomnia. So the correct answer for this question is option number 4. Let's proceed to the next question.